Thanks everyone for joining us today. We're here to have an exciting and scintillating conversation about one of the most compelling and possibly transformative technologies in the global fight to end malaria, which we all know is among the world's oldest and deadliest diseases. I'm Martin Edlund, the CEO of Malaria No More. Our vision, as you would guess from our name, is a world where no one dies from a mosquito bite. We've accomplished so much collectively in the malaria campaign over the last 15 or 20 years. 11 million lives saved, 1.8 billion cases averted, $2 trillion in economic benefits. But to achieve the ultimate goal, to end malaria once and for all, we need new and transformative tools. And that's what this Path to Zero series is all about. How we harness the latest breakthroughs from the cutting edge of bioscience to end humanity's most ancient disease. I'm delighted to welcome you to this, our second installment of the series. And before we get started, I'd also like to thank Oxitec for their generous sponsorship of this event. For the next hour, I have the privilege and pleasure of talking with two leaders in malaria innovation, Grey Frandsen of Oxitec and Helen Jamay of the Gates Foundation, whom I'll introduce properly in just a minute. We'll be talking about a technology that's moved from the pages of science fiction speculation to its very real debut as a viable and potentially game-changing tool in the campaign to end malaria. That technology is transgenic mosquitoes. Now, stepping back, we're at a really exciting moment, perhaps even a watershed moment in the malaria campaign. Investments made over the past 5 and 10 years by the US government, the Gates Foundation, and others have created a pipeline bursting with novel and transformative technologies. That pipeline includes a whole range of new tools, the potential for a highly effective malaria vaccine, monoclonal antibodies, next generation treatments and vector control. And we'll talk about many of these technologies over the course of the Path to Zero series. But none of these evoke as much interest, excitement, news coverage, or sometimes anxiety as transgenic mosquitoes. And that's really what the Path to Zero series is intended to help with, to demystify emerging technologies, to take seriously the complexity and the challenges they pose, and to make vivid the possibility they present for ending a disease that still afflicts hundreds of millions of the poorest people on the planet. Now, 15 years ago, we were able to successfully rally governments, the public, and donors behind one of the simplest and most cost-effective tools in global health, the long-lasting insecticide-treated bed net. In the space of 5 years, between 2004 and 2010, we went from around 5 million nets being distributed globally to 175 million nets distributed globally each year. And nets are a big part of the progress that I cited at the top of this session, the progress we've made. They remain an indispensable tool, a major reason that we're able to save nearly 1 million lives every year. Yet bed nets and current tools alone won't finish the job. We face new challenges that threaten to erode the effectiveness of bed nets and other vector control. The mounting challenge of widespread insecticide resistance. Of the 88 malaria endemic countries that provided data on resistance, 78 had detected resistance to at least one of the insecticide classes that are commonly used. We're also seeing new invasive mosquito species and new behaviors, like mosquitoes that bite in urban versus rural areas, mosquitoes that bite outside the home or even outside the typical nighttime window when Anopheles mosquitoes are known to bite and when bed nets provide protection for someone sleeping. So the upshot is we need new vector control tools that adapt to meet the evolving challenge of a changing vector. Hence our focus today on transgenic mosquitoes. So I want to introduce our panelists. I'll begin by welcoming Grey Frandsen to our virtual forum. Before being named Oxitec CEO in 2017, Grey held leadership roles in a long list of public and private sector organizations, including a series of startup companies he launched to help advance solutions to global challenges. He served as chief of staff for a new office within the US State Department and has served as an advisor to the CDC, USAID, Department of Defense, and other public sector entities on matters ranging from biotechnology to national security. Grey has also chaired the board of directors for an NGO, Pilgrim Africa, focused on malaria elimination and food security in sub-Saharan Africa, where Grey has focused much of his work for more than 20 years. Welcome, Grey. Also joining us today is Dr. Helen Jamay. Helen is an entomologist with a PhD in infectious diseases and more than 20 years of experience in the area of malaria prevention, research, and policy development. She spent more than a decade designing new next-generation vector control products for Vestergaard, the global leader in long-lasting insecticidal nets, before joining the Bill Melinda Gates Foundation in her current role as Deputy Director, Vector Control for Malaria. Helen spent many years conducting groundbreaking fieldwork in East Africa and is a passionate voice and advocate for innovation in the campaign to end malaria. Welcome, Helen.
Thanks.
We're excited to engage you both in discussion today. I'll get the conversation started with a few questions for our panelists, and then I'll turn to some questions from folks joining us live. So for those of you joining via Zoom, you'll have the option to submit questions through the platform. I encourage you to do so. And of course, we'll make this forum accessible for later viewing. So let's turn to our panelists and learn a bit more about one particular biotechnology in the fight against malaria. First to you, Grey and Oxitec. Transgenic or genetically modified mosquitoes is a technology that upends much of what we thought we knew about mosquito-borne disease. Over the weekend, I went back and watched the 1943 Disney film, The Winged Scourge. And that title really sums up how we typically think about mosquitoes. So in this 1943 animated film, it starts with a cartoon wanted poster. It says, "Public enemy number one, Anopheles, the malaria mosquito." Goes on to say, and I quote, "Wanted dead or alive for willfully spreading disease and theft of working hours, for bringing sickness and misery to untold millions in many parts of the world. Like all thieves and killers, she works best under cover of darkness." And the rest of the film goes on to show the seven dwarves in comical fashion hunting mosquitoes in all sorts of ways. But you and Oxitec are really flipping the script. You and your colleagues are even redeeming the image of the mosquito, branding friendly mosquitoes in your public health campaigns in Brazil and elsewhere. So, Grey, for those who haven't been watching this space as closely, tell us about how the science has advanced, what's changed in making it applicable today, including how Oxitec's technology works.
Yeah, good. Well, thank you for having me. Thanks for putting this on. You both are some of my favorite people in this community, and I love our conversation. So to have people listen in now, this is fun. Oxitec, just to say who we are a bit more, we're celebrating 20 years now, and I'm downtown Oxford as I log in with you, just down the street from where a small group of very sharp scientists determined that small alterations to an insect's DNA could generate some very significant benefits. Our entire platform is based on the idea that we use mosquitoes to hunt and find and mate with mosquitoes. It's very simple and very elegant. For us and for the company, we've developed a technology platform that inserts a single gene into the target pest genome. So in this case, we're talking about mosquitoes, that prevents the survival of female carriers to adulthood. That then gives us two things. One, it gives us highly efficient manufacturing, and two, incredible levels of suppression of a local target pest population. Very simple, very elegant, and tested around the world. We've deployed over 1 billion mosquitoes, well over 1 billion now, and have demonstrated exhaustively its safety, its efficacy, and really its scalability now. What most people don't know about us is the fact that Oxitec isn't just a company. We represent the work of thousands of people over the last 20 years, dozens of organizations ranging from Oxford University, where we got our start, but other universities, nonprofits, foundations like the Gates Foundation, all whom have come together to develop this technology platform, which will ideally provide a new set of solutions that are highly effective, targeted, environmentally sustainable, and ultimately scalable. So it's a wonderful representation of an incredible amount of work by a very broad and diverse coalition. So we're really proud of that. As we get into the discussion a little bit later, we can dig into a number of topics, especially around malaria. But I will say that as we celebrate 20 years, we are also celebrating the first time ever a transgenic mosquito has been sold to the public, where in Brazil, in the last number of months, we've begun selling our Aedes aegypti technology to the public. And I mention that here because when we talk about scalability of these solutions, Oxitec has taken a major step forward, and that is we've built the infrastructure necessary, we've generated the adoption and the acceptance for this type of technology, and we've shown the world that we're ready. And the exciting part of this discussion, Martin, is the fact that the need is incredibly urgent. There is a major need for new tools to combat malaria in this case, where the malaria fight is in some areas stalling. Climate change is pushing vectors into new areas, and we can't continue to do what we're doing and expect different results. And thus, we're working urgently to develop new solutions. And when we kind of think about the threat and the urgency of the challenge we face, like with Anopheles stevensii, the exciting part about this discussion is the fact that we've arrived, and we now are no longer talking about if these technologies can work, do these technologies play a role in the fight against malaria or dengue? That's no longer the question. The question now is, how fast can we get it to the field? And that's why I'm excited to have a discussion that's titled Ushering in a New Area, with the title that includes the word scalable as well.
Yeah, thanks for that, Gray. And I think it's, it may be part of the revelation for our listeners that we've really gone from this conversation being about science challenges to being about business challenges, to translating all of the good science and demonstration to actually impact in people's lives and communities. Can you tell us a little bit about the progress and capacities that you're building at Oxitec as you think about some of those business challenges, from production to distribution to logistics and partnerships?
Yeah, that's where I get really excited. And my background, Martin, I'm not a scientist, but for the 5 years I've been in this seat, the wonderful team here at Oxitec has focused on changing the mindset of the organization in general. We've gone from focusing specifically on the science at the lab, small scale field trials, to the rest of the stuff necessary to see these technologies deployed as solutions. Not science, not tech, but actual solutions that can generate and deliver exceptional value to end users. So we spend well over half of our time thinking about everything after a mosquito is developed inside the lab. So what are some of those things, and how do we kind of think about this? We start with the end. Oxitec has reorganized its entire purpose around delivering impact. And to do that, we have to develop and deliver exceptional products that add real value, real tangible value to end users. And to do that, we need to be an organization that's highly capable and highly flexible. And so we pride ourselves on now being a group of interdisciplinary professionals, ranging from, I'll be in the lab tomorrow, we'll be looking at robots, software development, shipping containers, logistics for moving things thousands of miles. We're solving for a lot of it. And to do that, we've gone to other industries to learn from the best. I think one challenge that we in the public health space have is we're very insular. We rarely think outside to others who have solved for things that frankly we shouldn't have to re-engineer. So for manufacturing, we're just 97% complete with a very large facility in Brazil. We went to the leading pharmaceutical engineering company to help us design a factory that is suited for exceptionally high quality and exceptional efficiency. Shipping, transportation, a big topic we spend a lot of time on because we need to be able to manufacture centrally and then push stuff out, to where it is needed the most in high quality, consistent fashion with a product that works. So we called Amazon. And we begin working with partners that are the professionals at moving goods from A to B. And I could go on. But the point is, we have transformed ourselves into a solutions delivery organization, not just an R&D, awesome, cool tech, awesome science, get a publication out type of place. And thankfully, we have a wonderful team that is continuing to press itself to evolve as our technology matures and frankly, as challenges emerge. COVID, stevensii, the spread of Aedes aegypti, all things that we have to continue to adapt to as we make these technologies scalable.
Thanks very much, Gray. And we'll get into some of those challenges as the conversation continues. Turning to you now, Helen. Obviously, none of this is news to you. You've been working in this space. The foundation has been investing in a whole range of technologies. How does this biotechnology fit alongside other transformative tools in the quest to end malaria? And what is kind of most additive or compatible about this technology amongst the others that you're investing in?
Thanks, Martin. Well, maybe to start off, just to kind of give a little bit of a very high level view of what we're doing on the malaria team at the Gates Foundation. We started our the strategy refresh that happened in 2018, took learnings from malaria and other diseases, including polio, and combined it with rigorous modeling to develop a strategy that's really intended to maintain and expand impact through the 2020s. And in parallel, develop tools, systems, and approaches that we need to ultimately achieve malaria eradication. And so this has led us to what we call the Pathway to Eradication strategy that's based on a set of key principles that outlines an agenda for eradication and provides a rubric for prioritization, preservation, and acceleration each year. And so we're including investing in ways to counter and stay ahead of resistance. So this is both in terms of drug resistance through new drug combinations and insecticide resistance through new molecules in indoor residual sprays or on bed nets. We're working towards shifting global and high burden country systems through subnational tailoring of interventions using data to drive decision making in a kind of closed loop. And we're also investing in the development of new categories of annual control tools, such as monoclonal antibodies and attractive targeted sugar baits in the vector control space. And what we consider to be transformation elimination tools that includes vaccines and genetically based vector control. As you mentioned in your introduction, vector control, including insecticide-treated bed nets and indoor residual spraying, has been the most effective tool to decrease malaria transmission. But the impact of these existing tools is really dependent on the continued efficacy of insecticides and repeated delivery campaigns to keep up high coverage. So while the insecticide-based vector control tools have helped carry some countries to elimination, they have imperfect protection. So we've already mentioned problems such as outdoor biting, but there's also imperfect use. There's growing insecticide resistance that you also mentioned, durability issues, and imperfect coverage levels. So this means that they are insufficient for getting transmission to zero in high burden countries. And so new, durable, and highly effective transformational tools for prevention are really going to be needed for eradication. And that's where tools like the second generation vaccines and genetic based vector control comes in. And gene drive is envisioned to be we call it a transformational tool, and it could be considered something that's a kind of gimmicky word, but we really think of it as transformational because of its potential durability and prevention of malaria reintroduction. It has the ability to self-spread to hard-to-reach areas independent of the health system. And it's also an area-wide intervention that doesn't depend on user behavior change. We see it as being very complementary, so definitely to be used along with other methods such as bed nets and seasonal chemo prevention. And there might even be a rationale for combining the different types of gene drive approaches that are being looked at in the future.
Thanks, Helen. And in my conversations with you and Philip and others at the Gates Foundation, it seems as though those investments in that pipeline is really showing promise. In fact, there's the vision that in the next 5 years, we may have a suite of tools that make that ultimate goal of eradication possible and could set up a big push globally, which is, of course, very, very exciting. You've talked about the broader landscape. Could you just double click into this area of genetic mosquito solutions and help us to kind of get clear on where things stand, the differences between the different categories of technologies?
Yeah, sure. So genetic control of insect vectors was really inspired by successes that started with the sterile insect technique that originated in the 1930s and 40s, that's been used to suppress screwworm, medfly, and tsetse fly. Sterile insect techniques rely on repeat inundative releases of sterile males, which then mate with wild females who then fail to reproduce. So unless the insect is completely eliminated, the effects are temporary, and it can be difficult to fund indefinite sterile insect release programs. It's not well suited to mosquitoes either, because they have larger populations and higher reproductive rates than traditional species for that type of approach. Gene drive is a phenomenon of biased inheritance. So basically the frequency of a genetic element increases even in the presence of a fitness cost that would otherwise select against it. So it could be an object like some kind of genetic element that biases inheritance in its favor, that can be used as a tool to affect population-wide changes in a way that a simple genetic modification with standard Mendelian inheritance cannot. The discovery, I'm sure everybody's heard of Crispr Cas9, the discovery of Crispr Cas systems finally allowed for precisely programmable gene drives that can be tweaked to target different genes via the use of specific guide RNAs. And the first mosquito gene drive was published in 2015. And since then, a number of proposed and engineered mosquito gene drives have proliferated, and crucial design issues such as the timing of Cas expression and the choice of target sites to reduce resistance have been substantially improved. So our kind of main idea of a gene drive is a low threshold self-sustaining drive. So basically this means that it can spread from extremely low initial frequencies, meaning you don't have to release many millions and millions of mosquitoes, and it continues to spread throughout interconnected populations without stopping. The widely publicized gene drives from Target Malaria and UCI Malaria Initiative conform to this type. So the major difference is that Target Malaria's gene drive creates population suppression, so targets the female fertility to decrease mosquito population numbers, while the UCMI type of gene drive is focusing on population replacement, so modifying mosquitoes so that they're unable to transmit malaria. And at the same time that we're supporting gene drive R&D, we're also looking at the development of non-driving transgenic anopheles for mosquito control. So both of the approaches that we're currently supporting cause offspring to be biased towards males, which temporarily decreases the total mosquito population, and particularly the female biting population. But they are eventually selected out due to high fitness costs. So Gray talked about the Oxitec approach, where their male selecting friendly mosquito uses a lethal element that's that only causes the males to survive to adulthood. And there are other approaches that we have in our portfolio. So using some natural elements, such as there's one research program looking at microsporidia. We've just starting an investment looking at Wolbachia as well. And I'm sure people have heard about the success of using Wolbachia and Aedes aegypti to drive that naturally occurring bacteria through a population to prevent the development of viruses like dengue and chikungunya in Aedes aegypti populations. And hopefully, particularly because that technology is much further advanced, there'll be a lot that we can learn from the scale up of those technologies. And of course, the Oxitec technology, which is further ahead. There'll be a lot of really important learnings, because Anopheles mosquitoes are very different to Aedes aegypti. So when it comes to figuring out how you do all of that scale up piece that Gray talked about, there's going to be a lot that we can then bring back to the gene drive space to help with the scale up of that technology.
Thanks, Helen. Really helpful to tease out those differences. And I've heard the Oxitec technology referred to as a self-limiting mosquito. Gray, anything you want to add on that before we move on to other topics?
No, I think that was very helpful. And I think we certainly celebrate the advancements being made elsewhere. And one thing that a few of us are working on, both of you are also supportive, is generating a stronger community of mosquito makers, such that those lessons can be transmitted. We're pioneers in a lot of ways. And I think the learnings we're generating, or the experiences we're generating, certainly hopefully can help others fast forward years even. So otherwise, good stuff.
That's great. So let's click into a specific example of a challenge where these technologies apply. And this is the story, and you referenced it briefly, but not everyone's conversant in all the scientific names of mosquito types. But it's an invasive species, Anopheles stevensii, a mosquito that's common in parts of Asia and elsewhere in the world, but has been seen in recent years in the Horn of Africa and now really the heart of Africa in Nigeria as well, which presents some new challenges, some different behaviors, and really put some pressure on the efficacy of current tools like nets. How concerned are you about these changes in Africa, and how do transgenic mosquitoes help to address these problems? Helen, maybe over to you quickly, and then Gray, for you to talk about how Oxitec is helping to tackle that challenge.
Yeah, thanks. We're extremely concerned. The possibility that this mosquito could lead to more urban malaria is real. This mosquito typically can breed in urban and rural environments, but it's found in very often in urban environments. It's a very rigorous and competitive mosquito, and it appears to be breeding in many of the same sites that Aedes aegypti would typically breed in. In one lab that I once worked in, there was Anopheles stevensii being reared next to Anopheles gambiae, and there was one mosquito that got into the Anopheles gambiae tray, and before we knew it, the entire colony had been taken over. So we really need to make sure that there's robust surveillance to track and identify where this species has spread. Unfortunately, there's still a lot of need for training in mosquito identification, particularly when it's a mosquito that people are not used to seeing. It's a mosquito that can feed on humans or animals, such as cattle, and tends to feed at night. But what we don't understand is how much behavior may have changed and how much it's already adapted to the African continent. We also don't know the extent of the spread yet. There was the recent report you mentioned that it was found in northern Nigeria. And we don't know if that's a result of spread from other countries or a separate introduction. Unfortunately, it's probably more likely to be spread from other countries. And so the great thing about the technology like the Oxitec friendly mosquito is that it's the mosquitoes themselves that search out others of the same species. So I'll maybe hand over to Gray to add to that.
Sure, that's great. Yeah, so Oxitec is developing a genetically modified or friendly Anopheles stevensii. We're really excited about it. This is our first foray into the malaria control space. We're doing so with great support from the foundation, with Helen's guidance. And we're really excited for a couple of reasons. One, obviously, the challenge is real, as you just mentioned. The eastern seaboard of the African continent alone presents a serious amount of risk as it continues to travel south. A few colleagues and I will be in Rwanda, Tanzania, Zanzibar, Kenya in the next couple of weeks, meeting with stakeholders on this very topic. And the fact that many of these big cities do not have the capacity for urban vector control. They don't have the tools, or they don't have the systems that would allow them to deploy an effective control effort. So, like Helen mentioned, we are very bullish on the utility of a friendly stevensii mosquito. And we're doing a couple of things right now. We're just now building a new facility in Djibouti, where we'll be carrying out our first field trials in the next year or two. We've partnered with the government of Djibouti very closely. They're very eager for an Oxitec mosquito to be deployed. And of course, more regionally, more broadly, Djibouti is seen as an entry point and a very important place where control needs to be gained. We're working on the strain itself, and we're making great progress. So for those not familiar with Oxitec, we run through a strain development program in the lab, typically here in the UK. Concurrently with that, we develop the rearing systems, the manufacturing systems, the product types for ensuring that it is maximally affordable and scalable when we need to, when we're ready for deployment. And great progress is being made on all of that. We're going beyond Djibouti, of course, in terms of stakeholder engagement. This will be one of the first GM mosquitoes deployed on the African continent. We have other efforts that were outlined by Helen active there as well. But we're spending a lot of time meeting and engaging stakeholders, listening very carefully and understanding how to build a very broad and inclusive stakeholder kind of coalition. I spend a lot of time on that, as do a number of our team members, and we're finding a resounding yes, please bring this technology as quickly as you can, given the nature and the size and the speed of the challenge. So good progress being made on this front. We're still a ways away from having a deployable tool. And as Helen mentioned, as other tools are failing or challenged, we certainly feel like this is going to be an important piece of the solution, part of the puzzle that's going to be necessary for success in combating this vector.
Thanks, Gray. And a couple of points jump out to me. One, a theme throughout is that transgenic mosquitoes are not a silver bullet, right? This is not a tool that in isolation is going to solve all of the emerging challenges that we have, but it may be a critical part of the kind of quiver of solutions that we have, and an especially good one at tackling emerging problems like the Anopheles stevensii example. It's worth also mentioning that these are not kind of marginal issues for the countries in question, the partner countries that you're talking about. As you look at the African map from Djibouti and Ethiopia in the east to Nigeria on the west coast and in Central Africa, that's a lot of territory. So if this is migration, that's a lot of really the heartland of the malaria burden that now faces this new vector and the new challenges that it poses. Nigeria alone accounts for 1/3 of global malaria deaths. Malaria is right there at the top of the leading killers of children under 5. So, these are in many ways the most urgent, the biggest scaled public health challenges faced in these countries. And yet, this is categorically new technology. There aren't necessarily systems for how to think about this either amongst the public or among policymakers and stakeholders and regulators. And we shouldn't minimize the complexity of that conversation. So let's spend a little time on those two issues now. Public acceptance is in many ways the heart of the issue. It's families and communities that bear the burden of mosquito-borne disease. It's families and communities who ultimately have such an important voice in deciding kind of what tools to use and whether mosquitoes should be deployed. Can you talk to us a little bit, Gray, maybe starting with some of your experience on how that conversation's going? What approach are you taking? What feedback are you getting? Where do we stand in that engagement with communities that stand to benefit?
Yeah, that's great. Great question, and a very important topic. It's really the prerequisite topic for discussions about successful deployment at scale. And we think about it in a pretty simple way. And maybe just for context, for those who don't know, Oxitec has been deploying mosquitoes for well over a decade in communities, multiple countries, multiple communities. And actually in every location where we have deployed mosquitoes, we have an exceptional level of public support. Sometimes the news headlines are a bit salacious and suggest otherwise, but the truth is that we generate trust wherever we go. So how do we do it? We start with trust. As an organization, it's our single most important value inside. We work to be a trustworthy organization so we can generate trust with those we seek to serve. We spend a lot of time and a lot of effort engaging where communities are, where they start in relation to this effort. We spend a lot of time listening and a lot of time engaging and educating at the door-to-door level, as well as at the kind of stakeholder ecosystem level. Now, we have 2 or 3 things we always do. We are always maximally transparent. And if you just flip over on a YouTube channel, on one of our YouTube channels, you'll find that we have done over 30 webinars open to the public in relation to our US efforts alone. It's incredible. I've never seen a biotech company, let alone a vector control company, do that. And I felt this was exceptionally important for us to basically have office hours. The door is open to anyone. We, again, just as one example, it's something we will continue to do. We're also maximally on. We're present. We're present in the places we seek to work. We integrate with communities, and we're basically always available. If we were, the convenience store of the vector control world, we'd be open 24/7. And I mean that. We have people around the world that are ready and active and engaging at all times, an incredibly talented team. I'll finally zoom back into Brazil and link it back to Stephen's Eye. In Brazil, we focus a lot on inspiring people to use these technologies. They are safe, regulators check, we engage, stakeholders want. Everywhere we go, we work in partnership, typically with cities or counties or states or nations. But how do we get people to get excited about adopting these technologies such that the chances for success go up? The way we think about it is generating a joyful experience in and around the product. Or as I share with my staff, nobody really wants to look inside Oxitec's, just add water mosquito product. Once you do it, it's a beautiful product. It's a beautiful box. You guys can see it online. But once it starts, mosquitoes start to come out. The stuff inside is pretty, it's not attractive. What we're trying to do, we spend a lot of time doing, is building the experience and the kind of the experience ecosystem outside of the box, such that people are engaging with us, with our partners, with our brand, with our messaging, can access information when and how they want it, and ultimately encourage high levels of adoption. And we're going to take that same approach to the Stephen's Eye campaign, on the African continent, especially where we are engaged in urban environments like we have been for the last 10 years in Brazil. We're bringing the exact, the pertinent lessons from how to engage urban communities, empower them to participate, activate them, sustain trust with them, and ultimately generate an excellent outcome. I think that we're uniquely suited for this bridge across the ocean, but bridge across to this additional species. But that's just some of it. Our website is chock full of stuff if people would like to see how we engage with people and communities. And I'd welcome anybody to reach out if they have further questions on it.
Thanks, Grey. And that transition from rural to urban is a compelling one and a different set of challenges. It makes me think, Helen, about, in the early days of scale up of long-lasting insecticide-treated nets, there was a lot of community engagement that needed to happen, a lot of engagement with governments. I had the great experience of living and working with Youssou N'Dour in Senegal and doing some of the first universal coverage bednet campaigns. And we had to think rigorously about how to encourage adoption, get people to use it each night. How is the Gates Foundation thinking about some of those issues of community acceptance and maybe bridging us to kind of regulatory pathways, engagement with governments to build awareness, to surface questions, to build capacity to tackle these issues?
Well, I have to say that the approach that Oxitec is taking really gets to the heart of what gene drive and genetic-based vector control approaches in general are trying to do, which is to prevent that biting from happening in the first place. And if you ask African partners what they're concerned about being bitten, they're concerned about getting malaria. And those get to the core of what this technology is intended to do. As you have alluded to, and you can see from the conversation so far, it's an extremely complex area with a lot of needles to thread. So we've been really thinking about both building the technology, but also building the platforms to help support the technologies as they develop. So we have investments with the Foundation for National Institutes of Health, who are leading expert consultations and coordinating and assisting partners to develop guidance on field deployment. The African Union Development Agency working on building biosafety, regulatory capacity and regional platforms in Africa. And another organization, Emerging Ag, is coordinating advocacy for gene drive research in key global fora. We also work with African partners on translating research results to policy to convene discussions around transformational tools that include gene drive in Africa. There's significant global and regional stakeholder engagement that's required to ensure that research can even continue, and also that communities are well informed about the science and the technical progress. Communities who will be the recipients of the technology are involved in the research even from a really early stage, and helping to define key areas of concern and questions that might need to be addressed, for example, through risk assessments. So people have all sorts of questions and concerns, as you can imagine. For example, what happens if people eat fish from a pond where the fish have been eating genetically modified mosquitoes? What happens if you remove an entire species from an ecosystem? All of these kinds of questions are taken seriously and addressed, and there's rigorous risk assessments that are being done to look at these kinds of questions and answer them, either using existing research or through specifically designed experiments that can investigate a specific area. Maybe I'll say a few words about the way that we're working with partners in Africa. The closer we get to a trial release, the more African partners need to be involved and engaged particularly in gene drive research and development. Not only do decisions about gene drive release need to be made by informed regulatory capacity and policymakers supported by local experts, but the programs themselves need substantial local capacity to carry out. So unlike a bed net or a drug, transgenic Anopheles mosquitoes aren't shelf stable and transportable over very long distances unless there are methods that eventually get created to help that happen. So they'll probably need to be reared and distributed by a capable local organizations located in the countries that they're serving. And that means monitoring is going to have to be carried out locally to enable programs to adapt as circumstances change. And so we're thinking about things like the intellectual property that's related to gene drive, making sure there is a license to operate so that it can be transferred or licensed to African-based institutions to ensure that there are gene drive programs that can be truly run by Africans. And African scientists need to develop the capacity to work with and engineer transgenic mosquitoes. And when decision makers turn to them for scientific input, they expect that the experts should have the hands-on knowledge and the deep understanding of the technology they're working, they're advising on. And there's a kind of chicken and egg problem. So the scientists can't work with gene drive material in their labs until the regulatory authorities have made a positive decision to allow the import of a gene drive mosquitoes. But the regulatory authorities also can't rely on local scientists to help with decision making until they have the experience and expertise. So we're trying to solve that problem with capacity building grants to African institutions so that we can develop the molecular biology and transgenesis skills that are needed to engineer and work with gene drive mosquitoes while addressing research questions that are useful in the near term. And then, of course, I mentioned working with the African Union Development Agency to build these collaborative regulatory platforms to help with reviewing dossiers and that should ease the decision making by sovereign countries.
Thanks, Helen. Sounds like a fascinating discussion, and I love the description of some of the kinds of questions that you get and have to think through and rigorously kind of demonstrate risks around. Grey, not to make a pun, but you have the gray hairs to show from your own long engagement with regulatory agencies. What are some of the lessons learned? What are some of the best practices from that work over time?
Yeah, it's great. It is a big topic, and it certainly is. I'm really thankful for what the foundation is doing by way of capacity building in places we hope to be in the future. There's the other end of the spectrum where certain regulatory agencies in certain countries are exceptionally talented and capable with significant capacity to carry out very robust risk assessments. And I say that having just, we're engaged in a number of countries right now on the regulatory front. Our US efforts are very well publicized by every media outlet. I think last year over 1,000 articles were written on Oxitec and the US. But all of that, kind of almost obscures how we work with regulators. And it's, again, pretty simple. We work to build trust with them. We base all of our work on the science and on facts. We're exceptionally collaborative. At least that's how I would characterize our efforts in spirit. We're prompt, we're fast, and we typically go way above and beyond as it relates to demonstrating both safety and efficacy profiles for our products. Some, again, for those who are just joining the discussion by way of the topic, the big questions on the regulatory front typically relate to, is there a regulatory body in a particular country that focuses on this? Many countries don't have a biosafety law or a department or division that handles this. That's a big, and the decision tree, that's a big one to tackle. Two, if there is an agency with agency and authority, do they have the capacity? Do they have the skill sets or the expertise? And to Helen's points, exactly. Three is, are there additional agencies one might have to go through? And we could continue on, but navigating all of that can be exceptionally involved. So in Brazil, Brazil's a very progressive, forward-leaning country as it relates to biotechnology, in large part because of agriculture. How big, basically the BT crop, the biotech crop business in Brazil is massive. And thus, there are some well-worn paths, and I'm really proud that we have been fully deregulated for both our Aedes aegypti mosquito and one of our first agricultural product, a fall armyworm, completely approved and deregulated, which is remarkable. In the US, we're a couple of years behind, but we're doing well. All of that to say, based on trust building, and a desire to build it, nurture it, sustain it. Regulators are just like other stakeholders in that they deserve our respect, our engagement, and in some cases, a little bit of debate here and there about how we can best demonstrate the safety and efficacy profile for our products, which we can do. Factoid, I think our EPA application required over 5,000 pages of material. And we have demonstrated empirically what happens when a frog eats our mosquito and something eats the frog, and it jumps around. We can show that. We can demonstrate safety in all of those areas, in part thanks to stringent regulatory requirements that basically have us do these things, which help then bolster our profile, our safety profile with those we seek to serve.
So Grey, you've answered one of the questions Helen raised. What happens when the frog or the fish eats the larva or the mosquito? One of the groups that's a part of these conversations, of course, is environmental groups. What are the questions, concerns raised by environmental groups? What are some of the maybe overlooked benefits of these technologies as we think about environmental implications? Helen, I wonder if you want to jump in first, or Grey, over to either of you.
Sure, yeah, maybe I'll jump in and then hand over to Grey. I think one of the big concerns is around the ethics of getting rid of an entire species that could happen with some of the suppression technology. it's really interesting when you speak with groups in the kind of Western Hemisphere versus groups in Africa. They tend to have very different concerns. The groups, as I mentioned, in Africa tend to be concerned about getting malaria and getting bitten. But there is a real real need to understand what happens whenever you change the dynamics in an ecosystem. You need to be able to have some idea of what's going to happen, because you don't want to have unintended consequences. And I think that one of the things that's really a really advantageous byproduct of the research that's going on in this area is the enormous amount of effort that's going into areas of research that, quite frankly, I don't think would be funded if Gene Drive wasn't being funded. So looking at in detail at food webs in the ecosystems where these mosquitoes live, looking at population biology and population dynamics and movement in details that we haven't had before. And if you look at the way that we control mosquitoes today, so for example, through the use of bed nets, there are areas of Africa now, area of Tanzania particularly comes to mind, where the use of insecticide-treated bed nets led to the local extinction of an Anopheles gambiae population. And it was replaced by another mosquito that is not quite as targeted by the indoor tools as the nets were. So it leads to slightly different issues and needing to think slightly differently about how you control that other mosquito. But it massively helped with the disease burden that one mosquito species was carrying. And I think we all recognize that there are a lot of malaria is very complex. There are many different vectors that are carrying malaria, and so removing one major vector doesn't necessarily mean that you remove all of the malaria. But if you can remove the main driver of burden so that you have a few secondary vectors that are perhaps easier to control, then you've gone a very, very long way to being able to get to elimination and eventually eradication that you wouldn't be able to get to without having that massive burden reduction through an approach like Gene Drive. I'll stop there and hand over to Grey.
Yeah, and I think another chapter of the topic then to cover on the environmental groups, advocacy groups, and the dialogue that happens kind of between a technology developer and the community it seeks to serve. For us, as a technology developer, we are certainly the subject of a lot of attention by advocacy groups that mostly in the US, mostly based in a certain area in the US. And they spend a lot of time thinking about us and finding ways to make things very difficult for us. So I certainly will just share that, reiterate what I shared earlier, which is we have excellent relationships with regulators, with stakeholders, with communities we seek to serve. And yet a lot of the stories tends to skew to the clickbait headlines that a number of publications will run based on what certain vocal organizations that call themselves environmental activists really are putting out there. So, but in our mind, there's two sets. There are environmental organizations focused on sustainability, ecosystem health, and we like them and they like us. And we typically have great relationships with professional organizations that have environmental stewardship as part of their mission. Why? We are a targeted solution. We have no impact on the environment. We eventually evaporate and leave the environment. It's a self-limiting technology that will ultimately not be present. Frogs can eat the stuff. And in some cases like Aedes aegypti, we can reduce the population and thus reduce the need for spraying broad spectrum pesticides that do have negative consequences on the health of an ecosystem. Not to mention the fact that many of these, like Stephen's eye or Aedes, they're invasive species. We don't want them there, nor do the environmental organizations want that disturbance in the ecosystem. So for us, the professional group of organizations working on environmental systems, they're really important. We engage with them, we have dialogue with them, we have support from them. And then on the other hand, the anti-GMO groups, let's just call it what it is, it's just going to be part of what we deal with. And as CEO of Oxitec, I have to deal with that, and it certainly isn't fun. But let's not let the true message and the true progress we're making get lost when big proclamations by anti-GM groups are made. I think that it's important for our entire community to hold hands, lock arms, and resting on sound science and open and engaging and empowering approaches, we can get these things to market. But we can't ignore it. We can't ignore that certain groups have a vested interest in raising money to fight these types of technologies like ours.
Thanks for that, Greg. And it's worth reminding folks that there are 3,000 some species of mosquitoes, right? And the technologies are targeting those that have the most devastating effect on people through these diseases. So we've talked a lot about the technology. We've talked a lot about the capacity. How close are we? Gray, maybe you can update us on kind of what's live and in the field, and what the timelines look like on the Oxitec side. Helen, as you think more broadly about the portfolio and gene driven mosquitoes, for instance, what do the timelines look like, do we think?
Yeah, okay, sure. Helen, I'll jump in briefly. So for Oxitec, as noted, we're now on the Aedes aegypti program. We're years ahead. And so we're in the market, we're selling to the market, we're engaging in commercial partnerships and activities, which, by the way, is a very important element to building an organization like ours that's capable of generating self-sufficiency as quickly as possible. I don't want to continue to ask Helen for grant funding in perpetuity, because that's simply not going to be a recipe for a sustainable impact at scale. The next step for us is a big agricultural pest. And that too feeds into the fact that Oxitec really is a technology platform that allows us to build multiple products. And that that's imminently, that's coming very, very soon. The Stevens Eye program, along with another malaria mosquito we haven't talked about, that's Anopheles albuminus, which is a Latin American malaria vector that we're still, and the foundation and others want to stamp out. We're still a few years away, Martin, and we want to get to field trials in Djibouti as quickly as as appropriate. I would anticipate that happening in the next couple of years. And as I mentioned earlier, we're paralleling efforts. Is that even a phrase? We have parallel efforts to ensure that we don't have to wait then for further engineering or innovation around rearing. We're trying to get all of that stuff wrapped up to be ready as quickly as possible. I wish I could say it would be next week or next month, but that's not the case for this particular technology. And Helen, of course, on your other portfolio items.
Yeah, so we're kind of between now and roughly around 2028, we're looking at new product categories that include monoclonal antibodies and the attractive targeted sugar baits that are currently going through, well, the first generation product is going through field trials to prove public health impact right now. For the kind of self-sustaining gene drive approaches, we're really still around a decade away from a first kind of fully fledged release, and then there'll be various iterations of smaller scale trials that'll be happening in the interim period. So we expect the Oxitec technology to be in the field first, which means that we'll be able to get a lot of learning from that we can then use to build on the other technologies. And it does give us, because we have this kind of conundrum of needing to build these platforms that I've talked about at the same time as the technology is being built. It does give us some time to continue working with partners to build the capacity that's required for handling this technology once we get there. So we have a decade, just under a decade, so lots of work to be done, but I think really exciting time ahead. And I think by the time we get to a self-sustaining gene drive release, we will have multiple different dual insecticide bed nets also in the field. So we'll be able to do a lot better proactive resistance management, both with bed nets and the multiple different indoor residual sprays that will be available, which should help. And then thinking through how we combine all of these tools, we are using continually modeling approaches to look at how we combine these different approaches, and that will be really important as we think through timing of a release and how the number of mosquitoes that need to be released, where you do your release points, that kind of thing.
Yeah, I think there's such a fascinating evolution of these strategies from kind of a one-size-fits-all approach. When we talked about bed net scale up, it was how do we get everybody sleeping under a bed net in malaria endemic sub-Saharan Africa, to much more targeted, layered kind of tools. Just a couple of thoughts to wrap us up. I think one of the critical things to remember in all this, you can get so deep into the science and the debate around these issues. It's worth remembering what's at stake here. It's worth remembering that a child is still dying every minute from a mosquito bite. This is at once our most ancient disease, but a very real day-to-day threat for billions of people around the globe. We're talking in technical terms, so I will mention DALYs. In public health, we talk about disability adjusted life years, a comprehensive measure of the cost of diseases. Despite all the progress we have, the latest numbers are that for every year that malaria continues to persist, we're losing about 44 million years of human productivity. That's the cost to the world of this single disease persisting. And so we've got to continue to innovate. We've got to explore the application of these technologies. We need to think rigorously. We need to do it in a stepwise way. But we also need to recognize that there's some urgency around solving these issues. So thank you very much to our two panelists today, to Grey Frandsen of Oxitec, Helen Jamay of the Bill Melinda Gates Foundation. I've learned a lot in this conversation. I hope our listeners have as well. So thanks to you both. Thanks to those who tuned in, and we look forward to our next Path to Zero technology series. And thank you again to Oxitec for sponsoring this discussion.
Our pleasure, Martin. Thank you. And thanks, Helen. Good to see you both.
Thank you very much. Bye-bye.